Bon après-midi. J'aimerais commencer pour pas trop tarder. Je m'appelle Elga Port. Je suis professeure à la faculté de foresterie, géomatique et géographie. Et ensemble avec Juan Carlos Villarreal, je présente notre conférence. Donc, j'ai le plaisir de vous présenter aujourd'hui le professeur Edel Pérez-Lopez. Et j'ai un petit peu mon cheat sheet avec moi. Parce que j'ai plus de rhume. En tout cas, donc, Edel est professeur et un expert en phytopathologie à l'Université Laval. Son parcours académique consiste d'avoir obtenu un baccalauréat en biochimie à l'Université de Havana, Cuba, puis son doctorat à Veracruz, au Mexique. Après, il a poursuivi deux postdoctorats. Le premier à l'Université d'Auburn, en Alabama, oui, aux États-Unis, et aussi le deuxième, donc, à l'Université de Saskatchewan, ici au Canada. Et depuis 2021, il est professeur maintenant à l'université et il est affilié aussi avec l'IBIS. Et sa recherche porte sur divers agents pathogènes végétaux et des insectes vecteurs. Et il s'intéresse beaucoup à la biologie des effecteurs. Donc, maintenant, j'ai le plaisir de vous introduire votre présentation. Merci et je me choisis.
Parfait, merci beaucoup. Et je suis très content de donner un séminaire ici. Et mon premier séminaire à l'Université Laval a été à l'IBIS, en ligne. Et dans ce moment, j'ai eu aucune idée de ce que je vais faire dans mon programme de recherche. Et maintenant encore, je n'ai pas aucune idée, mais on essaye. Je vais donner le séminaire en anglais, principalement parce que la moitié de mon laboratoire n'est pas français. Et je voudrais qu'ils comprennent qu'est-ce qu'on fait, mon propre laboratoire. Principalement parce que nous avons deux différents sujets de recherche. Mais, so yes, I arrived in 2020 with the COVID. It was exactly the same weekend that COVID happens. So I had to start my office in Saskatchewan in my room. But from that moment, we have established a lab that I want to believe we are a sustainable plant protection lab. We do many things, but the main goal is that to have a more sustainable agriculture, to use less pesticide from the side of the disease. Mainly we use the model of, no wait, this is still a problem. The light is this one, okay. So we use two different models. We use club root disease, which affect brassica crops. Canada is one of the main producers of canola, but also Quebec produces a lot of cauliflower, broccoli, different vegetables that are affected by this disease. But we also work with the insect pest, mainly leafhoppers. And today, I'm going to tell you more about the leafhopper side of the lab. And I love this side because it's one example of how we can go from very local and a small funding and a small project to a really pan-Canadian project. So I will tell you a little bit what we are doing here. So the story is back when I arrived. Valérie Fournier, she's the real entomologist in the lab, and she knew from my seminar presentation for the position that I was working with phytoplasma disease and with leafhoppers. So she told me that the growers, because she had been working with the growers for many years, they have been finding more and more leafhoppers. That's back in 2020. They were also seeing more disease. And for example, this is the green petal disease that affects strawberries. You can see that the name is because of the symptoms. The flowers turn green, and then there is no productivity. But they were also seeing more damage due to the feeding activity of the insects. And you can see here how the leaf turn yellow and the plants don't develop properly just because of the pressure of the insect feeding. Then, therefore, the growers were applying more insecticide, which is negative because here in Canada, in Quebec mainly, we do the auto QA. That's a really difficult mot en français. But we go, we pick up strawberries, and it's a social activity really, and it's very appreciated. So more insecticide, less access to the field when the time is the proper. So the disease is transmitted by leafhoppers. You can see here the cycle of the disease, how the insect feed, acquire a phytoplasma, which is a bacteria, a unculturable bacteria that can multiply in the insect, but also in the plant. And then the cycle continues, transmit the disease, and so on. Now, very interesting historical data. The first place where the maladie de la pétale vert was discovered in Canada was in La Pocatière in Quebec. So that's really cool in the 62. And I think that there was a history link to Quebec that I wanted to explore more. And like I said, strawberry production is a millionaire business for Quebec. We have more than 500 companies that, or growers that produce strawberry in the province. So we start with a little idea. There was a program in my pack called Innovación that was basically a little program of 3 years that you could develop knowledge about a subject that was still not a problem, like economic big problem. So we proposed to start investigating the diversity of leafhoppers. Now, why I didn't talk about the disease in that moment? Because it was not really a problem. So I said, okay, I want to work with the disease, but how can I get funding? So I went to the insect side with Valérie Fournier. So we start really small, 7 farms. We put yellow sticky traps every week, and we were able to collect the traps and then identify the insects. I just want to show you, this is in June 2022, and all the red dots were leafhoppers. So we had a lot of insects, and we also found a really beautiful diversity. They are amazing. I love leafhoppers. They are so beautiful, many colors. But the most interesting is that between 2021 and 2022, we collected more than 33,000 leafhoppers, and we identified all of them. So Nicolas, who is the student that did this work, have become an expert in taxonomy right now, so we have our own local talent. But very interesting, we found 118 species and 62 genera. So this was a way more than what was reported in the province before. Now, why is this important? And this is the work from abroad, and I'm combining different studies here. So this is important because when we did the modeling for only 14 of species that have been identified as vector of phytoplasma, we see in all the future scenarios that we are going to have more and more leafhoppers, but also more plants affected. Because what we found also is that these leafhoppers can feed of many different hosts. So there is the possibility that they can transmit many disease to different hosts. Now, what was happening in Quebec in terms of the disease? So here we collaborated with the Laboratoire d'Expertise en Phytoprotección, and they gave us the historical data in the last 10 years of how many growers were going to test the material and was positive for phytoplasma. So what we found was really interesting. This is not the number of plants infected, only the number of times a grower was going to analyze. And we found that there have been an exponential increase in the number of times that the grower have been worried with the disease. We also identified for the first time what was the molecular ID of this phytoplasma, what was the species, and it was a new species that was not reported before in Quebec. Now, Remember, we had data for every week during 2 years. So we asked to the growers, can you tell us when did you apply insecticide? So the growers gave the data and the product that they use. And what we did was a simple comparison to know if we were seeing a reduction in the number of leafhovers after applying insecticide. Very shockingly, there was no reduction with any products. And I think that we analyzed around 10 or 12 different products during 2 years. So something is happening that the insecticide is not controlling the leafhoppers. Now, we cannot say that they have insecticide resistance. That's something that we really have to test, and we are expecting a PhD student to work on this. But we can say that something is happening that insecticide is not controlling the abundance of leafhoppers. We were also interested in the microbiome. Remember that leafhoppers are vector of phytoplasma, but we still didn't know who was the vector of the phytoplasma causing the strawberry green parrot disease. So we analyzed the microbiome of the most abundant species of leafhopper present in Quebec, and no phytoplasma. So we still didn't know at that point who was the vector, but what we found is that the microbiome was changing depending the moment of the year. We analyzed June and August, and June is kind of the peak of abundance for this species, and August was when the species was starting to reduce the number of individuals in the population. And we found some changes, not critical, but we found that there was some influence in the microbiome for depending the moment of the year. So a little bit of a conclusion here is that we found changes in the abundance between the different years and the moment of the year. We found changes in the microbiome. We found also increase of the disease. And something that I didn't mention, but we also found, is that we identified 10 new species new for Quebec and 2 species new for Canada. So this was something really interesting. Now, there is also the bias. Sorry, is somebody else looking for that? No, not really. So we had a lot of chances to really find a new species. But what we propose is that leafhoppers in general could be a marker to study the impact of climate change in agriculture. Why? Because we saw this in so many different layers that this can be something that we can explore. This is not final, we're still working on this, but we were able to publish in this new journal and get the cover of February last year. Now, this kind of this kind of a study is really important because get a lot of media attention, and I wanted that precisely. I wanted to get a lot of media attention, a lot of momentum to be able to secure more funding and keep doing this study for a long term. Many of you are ecologists, so you know that 2 years is nothing in ecology. So you need 10, you need 15, 20 years to really have a strong, long-lasting conclusions. So we did that. With the Réseau québécois de recherche en agriculture durable, we secured 3 more years of funding to keep sampling leafhoppers in the same places, but now we double the number of fields that we were sampling leafhoppers. So between 2021 and 2024, we have identified more than 120,000 leafhoppers, and we have 120 different species. Now, consistent among the 4 years are these 2 species, Empuasca fabae and Macrosteles quadrilineatus. These are almost 80 to 90% of the capture that we get here. And the most interesting thing is that these 2 are migratory insects. So Macrosteles can still overwinter in Canada, but Empuasca is basically all migratory from South United States to Canada. Something that we did different in these new 2 years is to divide the insects in Empuasca, Macrosteles, because are the really, really abundant, and the rest we call local species. So this is very arbitrary because we really don't know if the 118 species that we group in here are migratory or not. This is something that we will have to work on. But consistent with the previous years, insecticides are still not controlling leafhoppers. So this was really important. We saw a little bit of reduction in Empuasca and the locals with this acetamiprid, but it's not really huge. As you can see, it's basically just slightly significant. But we still had one important question: how to control leafhoppers. We still don't know how to control leafhoppers. And that has become the focus of the lab, finding sustainable ways to control leafhoppers. So something that we observed in 2021 and 2022 is that there was natural parasitism in the field. So you can see here this bag called Tilasium. First time I see that, I say that name. We just wrote it in the paper, but haven't seen it. But this is a sign of parasitism. So what we did in 2023 and 2024 is to actively count the number of leafhoppers that had sign of parasitism. And the results were really interesting. You can see here in orange is Macrosteles, in green is Empuasca, and in blue are local species with Graminella nigrifrons is one of the local species. And you can see that most of the leafhopper parasites were Macrosteles. It makes sense in the sense that it's one of the most abundant, but Empuasca is also very, very abundant, and we basically don't see sign of parasitism or evidence. This is something that we still don't have the answer, but it's something that I like to point. Now, the parasitism rate is usually low. It's from 3, but it can reach 20%. Now, I have to mention that we are only seeing adults in this case. So usually parasitism happen in nymph, and I will show you a little bit later, but this might be the explanation. Still, 3% combined with an integrated pest management can have a real impact in the population and can help to reduce the use of insecticide. Now, the question: what is the parasitoid? What is the species? Who is the parasitoid in the field? And we did this with Joshua. We grabbed the Macrosteles quadrilineatus parasite. Then we extract the DNA, we sequence with Illumina, but we were lucky that in one of the traps also, one larva emerged from an infected parasite. So that was really cool. We had the larva, so we extracted the DNA. Was not enough to do Illumina sequencing, but was enough to do PCR and the CO1. What we found was really interesting. We found that we were able to assemble the mitogenome from the parasitoid coming from infected Macrosteles, and here is the CO1 in red of the larva, and it was the same. Remember, this was not the same individual. We grabbed infected Macrosteles, and the larva came from another one that we didn't analyze the odds. So what was the parasitoid? It's a pincer wasp. What is a pincer wasp? And here I have to show you the pincer wasps are in the family Drinidae, and the family Drinidae have different genus, but the one that was the closer one was Gonatopus. Now, Gonatopus is a very complex, taxonomically complex genus, and I love this, so I had to show you. What is the process that Gonatopus to feed or place the eggs? Because that's the interesting thing. Gonatopus not only put eggs in leafhoppers, it can also feed from leafhoppers. So this is the process. This is the species Gonatopus aboliniatus from Chile, I think, because this group is really good and it's from Chile. And start here, inject the venom, paralyze the target, the prey, and start feeding or putting an egg. So usually put the eggs in a larva, in a nymph, like I mentioned before, but it can feed from adults. Now, I sent Jordan, Nicolais from another time, but they do the same with the aspirator to the field because we needed adults to be able to identify who is the real species in the field. So Jordan went in September and started capturing, and it was really difficult. They are really tiny. So, but we got this beauty. So, so we got adults. We had 11 specimens, but we didn't know really if it's gonatopus or not because we needed to learn about the taxonomy. This is the first time I'm not a taxonomist. We are not a taxonomist group, but we collaborate with many people. And one of the people that we collaborated is in the National Collection for Hemiptera. So this is in Ottawa. We went to the collection and we start learning a little bit. There are some books that are only in physical, that you have to go in person and they don't give it to you, sadly. So we went there with our 11 specimens. We start learning how to read the taxonomy key because it's also very difficult. What are the different parts of the insect? And here, for example, you can see two of the species from the collection, Gonatopus contartulus and another one that I was not going to read. But as you can see, the ones that we captured were not completely similar, even at a glance, of the species that we found in the collection. So luckily, we had really good assistance to take a beautiful picture of many different taxonomical elements from the specimens, and we were able to identify at least few of the specimens as Gonatopus clavipens. So we had an idea, but we still are missing like 9 or 8. So with the collaboration of Florent, we had around 3 taxonomically different groups. But we then manipulate them a lot. You know, you were trying to identify, you move them, take pictures, and when we extracted the DNA from each individual, the DNA was extremely degraded. So no possibility to use any typical method, but we found a company that provided a degraded or ancient DNA library preparation. So we were able to go with that. We assembled all the 2 mitogenomes, but the most interesting is that we also were able to retrieve the CO1 from all of the, most of them, 10. And here are really interesting results. So you can see the Gonatopus immagrosteles, the larva, another the DNA that we sent again to the company, and a specimen 11, they all grouped together. So we had evidence that in the field we have something that is infecting macrosteles. That was really cool. The other cool result is that the specimen that we taxonomically identify was also a grouping, let me see if I find it, with Gonatopus clavipens. 196.3 is the branch. So that was really, really cool. So the taxonomy was matching the molecular results. And then we have this group here that is kind of divided into, that could be that taxonomically they are similar, but in terms of genome are different. Now, something interesting that I wanted to point about clavipens is that when we went to iNaturalist, we found that there were 2 records here, but they were records amplified by the project Barcode of Life. They were taken from a collection, but there is no data if they were really population that were active and described in the field. So consulting with the people in the collection, we are claiming that this is the official first mention of this species of Gonatopus in Canada. Something also really, really interesting that we found is reeds for Beauveria bassiana. So this is an entomopathogenic fungi that we found in several individuals of the parasitoid. And even when the mitogenome was not that good, it was very fragmented, we are really excited to know what is happening in this three level of a trophic interaction. Beauveria is a very well-known entomopathogenic biocontrol agent, so that's something that we want to explore in the future in the lab. So some message is that leafhoppers are a climate-sensitive model for sustainable pest control. This is something that locally we are going to continue to explore for at least 4 more years. So at the end, we are expecting to have 8 years of data if we don't get another grant to continue. I hope we do. We also found Drinid wasp active leafhopper that are active leafhopper regulators. So they are controlling this insect. An unexpected gonadopus diversity, we might have 4 different species in the field. And this was only with 11 individuals. So I'm excited to see what we are finding once we keep studying this. And a potential tripartite interaction. So this is something also that we want to explore. Is this isolate only affecting the biocontrol? That's not what we want. Is this isolate also able to colonize leafhoppers? Now, what is next? And this is very exciting. And here is Thomas, a new student in the lab. And I don't like to say no to a student. So when he came and say, can we try to have a colony of Drinidae in the lab? I said, try, why not? It's your PhD. So they have been collecting owls alive, and they are being keeping them alive to try to close the larva, and then the larva became an adult, and then have the full cycle register. And he have succeeded at some level. I will show you. So for example, this larva was closed from another leafhopper, sadly dying. But this is a process. We don't know anything about the biology of gonadopus. There is no study about the biology of gonadopus anywhere. So this will be really exciting. Then one of the other larvae that emerged was starting to do a cocoon. It was starting in a leaf. And even you can see here the eyes of the nymph. They was really excited. Sadly, it's not moving anymore, but we are still there. And then the last progress, and this was really exciting, is that he had another emerged larva, and then the larva created the cocoon against the falcon tube. So this is something that we are excited to, because maybe we don't need the plant for them to develop. And you can see that the larva was moving. Really cute. We also have to work in the video system, because we want to kind of have like a streaming or something of the emergence. So you can pay $1 to connect, and then we get money for funding. But so we are expanding our work for all Canada. So we were lucky to be successful in a recent a program called Sustainable Agriculture from NSERC, and we now have 5 different universities in Canada that are doing the work that we did here in Quebec. So we have people from BC, Alberta, Saskatchewan, Manitoba, Ontario, and Nova Scotia. So the LeafHope fever is around Canada. So this was with many, many partners. That was a lot of work, but I'm happy that we succeed, and this is happening right now. The team is also growing. We have Ariane, we have Alexandre. Alexandre, I have to put your picture, sorry. But many lab members now working on different levels of leafhoppers. We have genomics, microbiome, many things that are, because I cannot present today, it's a lot. But here are some pictures. We also implemented climate stations all around Canada. So we have real data from the field that we can now use for our modeling studies. We have, for example, well, here in the strawberry fields, we have now blueberry. You can see here it's in Quebec, here it's in BC. We have a low bush blueberry in Nova Scotia. The fields are completely different and the diversity is completely different. We have right now around 5 new species reported for Canada that were not reported before. That's around all Canada. Then we have canola, of course, because it's one of the main crops in the prairies. But this, interesting, these are canola from Quebec. So we also have some canola in Quebec. And we didn't stop there. We are now also working in leafhoppers in the Arctic. So these are insects that are very climate adapted. And the question is, why are they in the Arctic? Are they overwintering in those harsh conditions? Well, this is data from 2018 to 23 in collaboration with the project Sentinelle du Nunavik from the Insectarium à Montréal. And we have identified 21 genera, 32 species, 7 new species for the Nunavik, not for Quebec, but for the most north place in Quebec. And for example, this species is really interesting because it's known the Arctic leafhopper. So there is no weird that we are finding it, but it's interesting anyways to know how the population change every year. Now, this, for example, is in Cujuac, is where we were sampling this year in 2025. We don't get a lot. You can see that it's almost no vegetation, but we get still some leafhoppers, and we are really excited to know more about the microbiome composition of Arctic leafhoppers and the genomics comparing the population from the Arctic to the populations that we find here in the main, in the city or in Quebec. Now, what are we going to do with so much data? We are going to have a lot of data, and I've been thinking about maybe pest GPT. So this is something that maybe when I retire from 20 to 30 years now. But this can be, we have to integrate AI right now in our daily life. So maybe all this data can feed a generative AI that can guide growers and things like that. But this is, if you want to know more, we are organizing in November the Quebec Entomology Conference. I invite you all from 20 to 21 November here in Université Laval. And with that, thank you very much.